And finally, at the end of this talk, that's when you will have an opportunity to interact with and ask Professor Chandler some questions about her talk. Feel free to submit your questions throughout the talk in the chat function of Zoom. At the end of it, I will collate all of the submitted questions and I will present them, or I can ask you to unmute yourselves and interact with her directly. And with that housekeeping out of the way, let's just move on to our exciting Life from Lucy talk today. I consider it an absolute honor and a humongous privilege to host to you today at our Life from Lucy, Dr. Dawn Chandler, all the way from The Ohio State University. Professor Dawn Chandler is She is part of the Department of Pediatrics at The Ohio State University and at the Columbus campus in Ohio. But she's also a principal investigator who's affiliated with the Center for Childhood Cancer and Blood Diseases at the Wexner Research Institute. Dawn did her Ph.D. At the University of Texas Graduate School of Biomedical Sciences at Houston, and from the University of Texas MD Anderson Cancer Center, where she did her postdoctoral work. But amongst all of these things, which are of course great achievements, given that she has published in numerous high-impact journals as well, I have to admit that that the highlight of her career has to be me. You see, I was her PhD student. And I am absolutely delighted today to have her here with us, but most of all because she literally is the scientist who made me what I am today. She is more than just a PhD supervisor. She's a mentor. She's an amazing friend, and she's been a support for me all the way from the beginning of my career until this day, and I hope she will continue to do so. And so welcome, Dawn, to Lucy Cavendish College, and over to you.
Well, thank you so much, Aishwarya. That was one of the warmest welcomes I think I've ever received. I really appreciate it, and I am thrilled to be a mentor to you. And one of the best parts of our jobs as scientists, I believe, is passing down that torch to the next generation of scientists, and to being a mentor to multiple students throughout our career. So that's been really fun for me as well. It's been great to see you grow and achieve all that you've achieved. And I'm very happy to be here today. Thank you so much for the invitation. I will be talking today about ribonucleic acid, from ancient molecule to modern therapeutic. And I'll start off with a little bit of basics, because I believe we have a little bit of a mixed crowd that may or may not have a science background, but I will be getting into the depths of my research as well. So I just want to make sure that some of us are all on the same page so that we can at least follow along in broad strokes as we get started here. So if I can make my slides advance, we'll be in good shape. Okay, so if I asked any non-scientists on the street what DNA is, most of them will know. They might not know it stands for deoxyribonucleic acid, but they will know that it's the genetic information that's encoded within the cells of an organism. And so the chromosomes, the DNA is organized into chromosomes within our nucleus of our cells, which is the center core of our cells in each cell. And they are made up of 4 different bases, A, C, G, and T. And it's a double-stranded helix that we've heard about since the days of Watson and Crick. And so that information is very important because it really tells us, or tells the cell, what it will become and what the organism it will become. So in really big picture, it tells you whether you're going to be a mouse, a frog, a dog, or a human. So that genetic code really tells you what kind of being you will be, and what color hair you're going to have, what color eyes you're going to have, how tall, etc. And also, amazingly, it also tells each cell what its identity will be. So whether you're going to be a nerve cell or a muscle cell, this genetic code is required for information for each cell to do its job and to become morphogenetically distinct as independent cell types. So how does that genetic information do its work? How is it passed down? So it's passed down from generation to generation. I think people know that it's inherited. How does it work in the cell? So that's the central dogma of molecular biology. Here again is the DNA, which is a double-stranded helix. This DNA is copied almost identically into an mRNA. And mRNA stands for messenger. It is the messenger. It takes the blueprint from the library to the factory so that the proteins can be eventually made. RNA is also made up of 4 bases, although instead of being A, C, G, and T, it's A, C, G, and U. These things are triplet codons, so each triplet repeat here codes a particular amino acid, and those amino acids are added in strings to make proteins. And the proteins are the molecular functioning part of the cell. They do the heavy work in each cell. So importantly, messenger RNA or mRNA, all of you probably have heard of now, since probably most laymen didn't know what RNA was. It's ribonucleic acid, much like DNA. It's very similar, but it's the messenger, the mRNA. And since 2019 and 20, with the onset of COVID, almost everyone has heard of mRNA. COVID is a 19 caused by the SARS-CoV-2 virus is an RNA virus. And importantly, the mRNA, one of the mRNAs that encodes a single gene from the SARS-CoV-2 virus is important for the vaccine that most of us have received, the mRNA vaccines. So from being DNA's primordial cousin to being on the front pages of every newspaper, mRNA has had an illustrious career in the last couple of years as an important therapeutic. So how is RNA processed and how is it made? It's copied from the DNA, but it's not a perfect copy. What you get from the DNA is not what's going to make the protein. It has a lot of extra sequences. So what you, or extra bases. So what you see here are these boxes. These are called exons. These exons are the sequences that you want to keep that are important for that message, that are important for making that protein. These lines in between the boxes are called introns. Once they need to be removed in order to be able to read the transcript or to pass down that message. So the DNA makes this RNA that's much longer in most cases than it needs to be. It needs to be extensively processed. There are a number of different processing steps for this RNA. And today I will be talking to you about splicing. And so splicing is the removal of these introns so that these exons can be ligated together to make the complete message to make the protein that you make. But what's even more fascinating is that each transcript, the mRNA that's made from the DNA, can be processed in multiple different ways. And these multiple different ways can make multiple different proteins. For example, in this instance, you can include all exons to make this really long protein that has one function, and you can skip exon 3 to make a different protein, or skip exon 4 to make a third protein. In this case, alternative splicing makes 3 different messages, 3 different mRNAs, that are then made into 3 different proteins that can have wildly different functions. They can negatively regulate each other, they can have slightly distinct functions, or they can, one can be non-functional altogether. So this is a very nice way to expand the genomic diversity and information that you can get from DNA. So how does RNA processing work, and what does that mean? So RNA processing must be very precise. In this case, we have a 4 exon gene with the exons, let's eat grandma. And in one case, when RNA splicing occurs, you get all 4 exons being included, and you have a sentence that reads, let's eat grandma. It indicates a very nice afternoon with the granddaughter and a grandma having a picnic, perhaps. And this is when exon inclusion occurs and all exons are recognized. However, if the cell tells the transcript to be processed in a differential way, following these top lines here, you might get a completely different outcome. You can get exon skipping. And now this important exon that contains the comma is left out, and the sentence has an entirely different meaning. Let's eat grandma. Let's eat grandma. So now you have a different picture of a mean wolf ready to devour grandma. So as you can see, splicing can make completely different outcomes as you can in this sentence. You can have a very pleasant outcome from the correct splicing, but if splicing does not occur properly, you can go into a disease state. And so that's what I study in my lab, and that's what I'm going to be talking about today. So in my lab, I talk about working on insulin receptor, and insulin receptor is differentially spliced in cancer. Here, I'm just showing you 3 exons that are the important part that are being processed differentially with insulin receptor, and that's exons 10 through 12. Exon 11 is a very small exon. It only contains 36 of those bases, those ACGs and U's, and when it's alternatively spliced, those 36 nucleotides are missing. So it's a 12 amino acid change in the protein. Not a very big change in the protein, but these proteins have vastly different functions. So when you have IRB splicing, you have full-length splicing, all exons are included, and you have normal metabolism, a very happy outcome, much like Little Red Riding Hood having lunch with her grandma. However, when exon 11 is skipped, you get a short RNA. The comma is no longer there, and it completely changes the meaning of this protein. This happens in cancer and under hypoxic conditions. So cancers grow so quickly that they don't get enough blood from the blood supply, and they get hypoxic. They don't have sufficient oxygen. And this hypoxia causes them to change their splicing. And that splicing changes, those splicing changes help to drive the cancer phenotype. So in one case, you get differentiation from the production of this IRB signaling isoform. But in the other case, you get an IRA, which now binds to a growth factor that's secreted by the cancer cells. And this growth factor portends growth and facilitates proliferation, cell motility, and angiogenesis to feed that cancer with blood and oxygen. So now you have a pro-tumorigenic isoform that allows cancer to occur, and you have the wolf, and you have the bad outcome from the splicing change. So how is insulin receptor spliced in pediatric cancer? So I study a pediatric cancer called rhabdomyosarcoma, which is a muscle tumor. And if you look at muscle tumors, a normal muscle, you can see full-length splicing. This larger band here on this gel indicates a band that includes exon 11. However, you also see a small amount, or about half of it, is this exon 11 skipped event. So a smaller band that lacks this exon 11. You can see these 2 bands very clearly. This is a normal muscle here. However, if you look at these cancers, this is from 2 different types of rhabdomyosarcomas. On the top, we have embryonal rhabdomyosarcomas, and on the bottom, we have alveolar, which are 2 distinct types of cancers, and these are from actual patients. You can see we have a dramatic shift to this bottom spliced isoform, a dramatic shift to the wolf, or the cancerogenic isoform, cancer promoting isoform. And so you can see that in both these cancer types, alveolar on the bottom and embryonal here, and that's true also in cell lines grown in tissue culture. And you can see in normal muscle, again, mostly full-length insulin receptor and just a little bit of skipped. So the alternatively spliced insulin receptor A isoform is prevalent in rhabdomyosarcoma patients and cancer cells. So we wondered if this might be driving cancer, and if a switch in splicing might be important as a therapeutic avenue, and if we'd be able to achieve this. So again, this is just to drive home the fact that pediatric cancer cells really show this shift in splicing. You see a really dramatic increase in insulin receptor spliced A, which is a skipped isoform in rhabdomyosarcoma, like I mentioned, and osteosarcomas, which is a blood tumor in children, a bone tumor in children, and Ewing sarcoma, a different kind of bone tumor in children, and clear cell kidney sarcomas. So you can see that this is a very important alteration that's prevalent in numerous different kinds of pediatric sarcomas. So why does the splicing of insulin receptor change? I already gave you a hint about this in my introduction slide to insulin receptor. Hypoxia is one of the hallmarks of cancer, hypoxic conditions, low oxygen conditions, because the cancer cell grows quickly and outgrows its oxygen supply from the blood. And we have a hypoxia chamber that mimics this cancer's environment. And what you can see here in and these cells that are grown under normal, these are again cancer cells, which have already increased their splicing of insulin receptor A, or skipping of that exon. You can see that in hypoxic conditions where that oxygen is depleted even further, like you see in cancer, you see even more of this insulin receptor A, or skipped isoform. And again, that's shown here graphically and quantitated over numerous samples. And if you look in databases that are available from sequencing from patient samples, you see that hypoxic conditions, when HIF-F1 expression is up, you also get expression of insulin receptor A. So we wanted to understand how the splicing was being regulated so that we could interfere with splicing for a therapy. And to do that, we had to make a mini gene. So instead of studying the context of these genes in the genomic context, which is multiple kbs long, we made a mini gene that contains only the 3 exons of interest with the exon that's being regulated in between. We can transfect this into cells as a surrogate of splicing regulation. And we identified, with the help of others, that Nick Webster and others that have looked at this splicing of this gene in the past, to identify upstream a CUGBP1 binding site and downstream a muscleblind-1 binding site. And these are sites where RNA binding proteins can bind to the mRNA transcript to regulate processing of this exon 11. And when we made mutations in this mini gene, you can see here the mini gene splices just like the normal gene. In normal cells, you get a little bit of IRA. In hypoxia, you have a push towards IRA. So the mini gene splices much like normal cells do. And when you make a mutation in the CUGB binding site, you get restoration of this normal, or Little Red Riding Hood, nice grandma afternoon isoform. So the normal healthy isoform, you can abolish insulin receptor A, the one that promotes cancer. So we thought maybe this would be a nice target for therapy. And so we could actually turn this bad isoform into a good isoform. So how do we accomplish that? So there is a technology called antisense oligonucleotide technology. And basically, you can take these elements that we've identified in the RNA and target them with these ASOs with anti-parallel sequences. So the same way that DNA, the 2 strands of DNA are hybridized, you can make a very short oligonucleotide made of DNA or RNA that can bind to the RNA transcript, and it can block binding of the protein then. So you can actually, if you have a positive element that facilitates inclusion of an exon, you can block that with an inhibitory antisense oligonucleotide, or SSO, splice switching oligonucleotide, and you can induce exon skipping by blocking this positive element. In the converse, if you block a positive element, you can induce exon inclusion with another positive acting splice switching oligonucleotide. And that's what we would like to do for insulin receptor, is really re-include this exon 11. So we looked to see if we could use an SSO targeting our CUGP binding site. So how do we do that? We designed an oligonucleotide walk of 21 base pairs of nucleotides that were anti-parallel and complementary to the entire CUGBP1 binding site that we found upstream of exon 11, to a CUGBP1 binding site that we identified within exon 11, and to the positive acting muscle-blind site that we found downstream. So you would expect these oligonucleotides to force inclusion of this exon, and these oligonucleotides to force skipping of this exon. And we did this in collaboration with Ionis Pharmaceuticals. And so we were able to find oligos that did what we expected. We have ASO 55 that targets the CUGBP1 binding site, and ASO 23 that targets the muscle-blind binding site. And you can see those results here, where the 23 nucleotide, 23 ASO, switches it completely to the damaging, cancer-promoting IRA. However, the ASO at 55, which targets the CUGB1 binding site, switches it almost completely to the normal, homeostatic promoting, metabolism promoting IRB isoform that includes insulin receptor exon 11. And so this is just an example of that walk that we did. In this intron, you can see that we see different varying degrees of inclusion, and that much like oligo 55, exon oligo 88 also does a nice job of including that exon. However, when we target the same CUGB binding site that happens to be within the exon, those do not work. And that's probably because the exon is so small, and now we're interfering with the consensus binding splicing signals on either end of that very small exon. So these are not good drug candidates, whereas exon, the intron ones, 88 and 55, are good drug candidates. And that's just here to show, again, to really knock it home, that we've identified oligos on both sides of this exon 11 exon that target this positive, the negative element, so a positive acting ASO, or a negative acting ASO that targets the positive element here. So what can we do with this, and how is this going to be helpful to us? So ASO treatment restores insulin receptor splicing to INRB. So if we can treat the cells, we can treat the cells with insulin receptor. You can see here, these are 3 different rhabdomyosarcoma cell lines. So these are cell lines that have been taken from patients and can now be grown indefinitely in tissue culture dishes. And you can see when we treat with ASO 55, we see this nice dose response induction of INRB in the 3 different cell lines that we do not see with the non-specific antisense oligo in any of these. And you can see that really nicely graphically shown here. So we were able to restore splicing. So will this help us with the disease? It's not as black and white as it is in the other, in the mini gene example that I showed you before, because we have a switch, but it's not 100%. So is this going to be therapeutic? That's the question we really want to ask. Can we get enough IRB and get rid of enough IRA to really have an effect on these cancers? So we treated cells, again, rhabdomyosarcoma tissue culture cells.
Just need to, my mom's texting me to.
And we checked for proliferation. You can show these are, you can see here that these are proliferation curves. You can see that we have a nice, rhabdomyosarcoma cells grow very well. And when you treat these cells with ASOs, you get a nice induction of IRB. You see hardly any in these cell lines without ASO. You see a really nice induction here. It can really decrease the rate of growth of these cancer cells. And when we take the same cell line and transfect with an insulin receptor that cannot be changed by splicing, you can see that we no longer can block that anymore, showing that our ASO is specific and really acting through splicing of the insulin receptor. So we know that ASO can change splicing and it can change proliferation rate. Two things that are important for these cancers to grow. And so one of the things that cancer cells do also is they migrate and they cause metastasis, and they can really, and this is shown in the laboratory, when you can make a scratch across a plate, the cells can migrate through that scratch quickly if they're cancer cells, and non-cancer cells do that very slowly. And so you can see here that with these same rhabdomyosarcoma cells, if we treat them with a splice switching oligonucleotide, you can see that the cancer cells that are treated with a non-specific oligo, where you do not see any change in splicing, close this gap very quickly, and they travel very fast across this distance. However, when we treat cells with antisense oligonucleotide, we were able to prevent that migration and slow down that migration in cancer cells, another hallmark of neoplasia that you'd really like to get rid of. And this is again showing that we can close that gap in normal cells, in cancer cells very well, that have not been treated, that have been treated with normal oligos, and not close it when we treat the cells with ASO. And that this is again specific, and that if we transfect with an IRA GFP that cannot be changed the level of splicing, so it's a cDNA, we no longer see this difference. So how does SSA the splice switching oligo treatment affect cancer cells? So cancer cells produce MMP9, which is a a protease that really facilitates metastasis. And you can see here that MMP9 goes down very dramatically when you induce with splicing with the ASO or splicing with oligo. So in the cancer field, because so we are able to do that by changing splicing IRB here back to IRB. However, there is an antibody that blocks IGF-1R and blocks this arm of the growth and proliferation by signaling of the IFR1. So if we are able to block both IRA and IGF-1R, are we able to increase or improve this efficacy? And what you can see here is that you actually can. So combination of SSO and the IGF-1R antibody shown here, blocking this receptor, really does make all of these TGF-beta, which is a growth factor, and other metastatic promoting signaling pathways go down. So not only is MMP9 now affected like it was by the splicing oligo, but the combination effect of this IR antibody as well as the changing of splicing now blocks both of these signaling conduits down this pathway, and we get this really nice change in multiple pathways. So the question is, does this affect angiogenesis and then affect the viability of the tumor? And so again, we're going to be doing this with both SSO and IGF-1R antibody. So here we've shown an angiogenesis assay, which is a surrogate assay to show branch formation, how vessels are formed in cancer cells in an in vitro construct. And so in this dish, which are only treated with PBS, we do not see any branching. So the cells are unable to make these vascular-like structures. And when we treat the cell with VEGF, you can see lots of branching, and you can see this vessel formation. And when you treat the cells again with non-specific antisense oligo that does not change splicing, so that maintains the IRA, you still see lots of branching. So you see no change here. However, when you treat the cell with ASO 55, you start to see decreased branches and that less fidelity there. And this is even more exaggerated when you treat with both ASO 55 and IGF-1R. And that's shown graphically here in this last bar, where this is really a combinatorial effect. I'm just moving my windows around so I can see my slides. It's one bad thing about Zoom. So this works in a tissue culture dish. We can slow the growth of cells. We can slow what looks like vascularization of the tumor in vitro. But is this going to work in a patient or in a tumor? So we turned to mice to check this out to see if we can get it to work. And so we treated mice. We made tumors in mice by injecting immunocompromised mice with Rh30 cells so that they can grow a tumor. And then we treat those tumors with either the non-specific oligo or the splice switching oligo. And we harvest the tumors and look for angiogenesis and signs of tumor progression. And what we saw was a really nice, number one, induction of splicing, of induction of IRB or exon 11 included transcript. And we see decreased angiogenesis when we have IGF-1R antibody plus the SSO, and that's just quantified here really nicely. So this looks like it's something that can be used in vivo to slow the progression of tumorigenesis. So what is the mechanism of this action? And is it working through phospho-AKT pathway, which is the pathway that promotes proliferation, cell motility, and angiogenesis? And this just shows here that yes, that is the case, is that when you have SSO 55 shown here, and the IGF-1R antibody, that you have increased resistance to phospho-AKT and this growth signaling pathway. And again, that's just shown graphically here. So with this very primordial molecule, the mRNA, we've been able to target it to treat cancer therapy. And so what we envision happening is using a combination therapy for this particular growth factor cascade by blocking the IGF-1 receptor with antibody and changing the insulin receptor splicing back to insulin receptor B so that we can slow down proliferation, cell motility, and angiogenesis and really promote metabolism rather than cancer and really have that picnic lunch with grandma rather than having the wolf by controlling splicing. And this is opposed to what you see in cancer. We really have this predominance of insulin receptor A and signaling through IGF-1R to really have proliferation, cell motility, and angiogenesis. So this is what we envision happening. And so in cancer cells, you have all these upregulation of these cancer hallmarks. And in tumor cells where you have splicing restored to the normal phenotype, you have decreased cancer cell hallmarks and hopefully a regression of tumorigenesis. So you have application of ASO that shifts the splicing to insulin receptor B. And so we still are left with numerous challenges to be able to deliver the SSOs to tumor cells in vivo and are more complex splice variants targetable. So this is a very simple splice change with a single exon that's important for this cancer phenotype alteration. Some transcripts are much more complicated. I'm going to tell you a little example about that coming up if we have, I think we have enough time. I won't get through all of it probably, but I'll talk about our more complex splice variants also targetable with ASOs and can maybe used to change a multitude of different splice variants. And so I'm going to switch now to regulation of MDM2, which is a transcript that is very near and dear to Aishwarya's heart. She worked on this in my lab many years ago, and we're still working on it today because it's very complicated. So much different than the insulin receptor splicing that I just told you about. MDM2, which is the murine double minute gene, which is a negative regulator of the prototypical tumor suppressor P53. It is very complicated splicing. So it has 12 exons. And 8 of these exons can be excluded together to make a very short isoform. So in normal cells, again, you have exons 1 through 12 being included. And this full-length MDM2 protein negatively regulates P53 and keeps P53 in balance. It's a tumor suppressor gene. It's not needed unless the cell's under attack or it has DNA damage. And so MDM2 really keeps those levels in check. But in response to damage, when you do need P53, one of the ways it's turned on is through the splicing event, where you get skipping of exons 4 through 11, a short form of MDM2 being made. And this form now binds to the full-length form of MDM2 and negatively regulates it. So it has a polar opposite effect. It's not a change in signaling, it's a complete dominant negative. And now you have P53 turned on, and it can respond on the insult in the cells. So how would this be important for cancer therapy? P53, as I mentioned, is a tumor suppressor gene. It's the guardian of the genome, and the cells are protected when P53 is turned on. However, in normal cells, P53, which I've depicted here as the soldier or nutcracker, is here asleep with Claire in the Nutcracker Ballet. And MDM2 turns off P53 in normal cells, but when it's not needed, MDM2 keeps it off, and so the guardian is not required. However, in some cancer cells, MDM2 is overexpressed to such a degree that P53 can't be turned on, and now these cells are no longer protected. So it's sleeping so hard because MDM2 is keeping it turned off so strongly that you can't get any tumor suppression, and you really need to be able to turn it back on to make it sensitive to different chemotherapies and to have its tumor suppression activity. So our goal is to induce MDM2 alternative splicing and to turn off MDM2 and to reactivate this guardian of the genome. So to do that, we wanted to understand how MDM2 splicing is being regulated, and can we target it with these same kinds of antisense oligos to force skipping and turn on P53. So that was our quest. And so to do that, we found that MDM2 is regulated by genotoxic stress to turn on the short form. Again, this long form, this big form running on this gel is the full-length MDM2, and this is a skipped form of MDM2. It's induced very nicely by UV and cisplatin. So we can really use this to study the regulation of MDM2 and understand what molecules are important for regulating it, what sequences are important, and what sequences we could potentially target with antisense oligonucleotides. And so again, we made a mini gene. This time, we didn't include all the exons that are important because there's too many exons that are being skipped. We included the terminal exons of the splicing event and one intervening exon. And surprisingly, this very small mini gene that lacks most of these skipped exons still is responsive to damage. So in normal cells, when you transfect this mini gene in, you get full-length splicing. And when you induce stress, again, you see this induction of skipping, much like you see in the endogenous gene that I showed you earlier. So we have this mini gene that allows us to really characterize the splicing events. And what we've found using that mini gene is that there are two important positive regulators here that bind to SC35 and SRSF2. That binds at 165 and 213. And you can see that when we make mutations in this binding site, we can get rid of this nice induction. So here you see in the normal UV, you get an induction. However, when you make mutations, you have this skipping even in the absence of damage. So we really have found these positive elements that might be targetable with ASOs. And so we tried targeting these elements with antisense splicing oligonucleotides, splice switching oligos, and we targeted both of these elements at both ends of this exon. And you can see here, the non-specific SSO, again, does not alter splicing, but the SSOs 1, 2, and 3 that target two different elements are really good at promoting now skipping. So these SSOs are different than the ones I talked about earlier. They target a positive element and they induce skipping. And this skipping can be induced pretty well by SSS1 targeting this element, but this element is really the gold mine here. It targets so well and gets almost 100% induction of skipping in this mini gene context. So now, does it work in endogenous gene? We have included SSOs targeting the endogenous MDM2, and we get induction of MDM2 and induction of P53. So that downstream target, we can get splicing changes and we can get P53 changes, as well as changes in the downstream target genes of P53. And that's just shown really nicely here with this really clear induction of MDM2 alt1 that correlates with an important apoptotic response gene that's activated by P53, which is turned on when MDM2 is turned off, PUMA, and that's just shown here. So this is just showing that this SSO can work by targeting a single element in a downstream exon in MDM2. And so can we get this into cells in vivo? So we thought about using lentiviruses, which can express cells in vivo. And we cloned this SSO into a lentivirus, and it's now making an siRNA, which is a short, an interfering RNA that we proposed could act the same way of acting on splicing. And when we express this lentivirus in our rhabdomyosarcoma cells, you can see very nicely here that with a control lentivirus, we only see full-length MDM2, but with the SSO3 containing lentivirus expression vector, we get this nice induction of MDM2-alt1. So it looks like this might be a therapeutic avenue for reinducing P53 activity. However, lentivirus isn't very clinically relevant because you can't treat patients with lentivirus because it integrates into the genome and could cause secondary mutations that might be dangerous. So this is really nice in a laboratory setting to show proof of concept, but it cannot be used in a clinically relevant way. So we wanted to think of other ways, how else can we deliver this oligo for a therapeutic benefit? And so most of you are well aware of the mRNA vaccines that have been used to treat COVID and to vaccinate the population against SARS-CoV-2. And so the way that these vaccines are made is that a lipid nanoparticle encapsulates an mRNA, the messenger RNA from the virus, and it delivers this mRNA to your cells, and your cells start making protein off of this based on this construct. So we thought, if you can deliver mRNA with a lipid nanoparticle, surely you can deliver an siRNA with a similar lipid nanoparticle called an extracellular vesicle, which is a nanoparticle that's made by your cells itself. So we thought maybe we could do this, and maybe we could target and deliver these splice switching oligos in this manner. And so we hijacked the lentivirus system, because the lentivirus, lucky for us, makes extracellular vesicles as part of its natural life cycle. So it expresses RNAs and proteins, and it makes them expresses them in multivesicular body within the host cell, and then it dumps these extracellular vesicles that contain this cargo into the extracellular matrix and delivers it to other cells. So we thought, can we make our cells express lentivirus, make EVs that express our SSO, and deliver them to cancer cells? So we did some tests with some proteins that express red, so we could see them in cells, and indeed we can deliver the red cell, the red expressing gene, to these cells using extracellular vesicles. And so next we thought, can we deliver it to tumors? We can do it in cells and cell culture. Can we deliver it to tumors? So again, we are delivering this this red with extracellular vesicles into a mouse, and you can see very nicely that this tumor on the flank of this mouse is now expressing red, and that's because we injected it with extracellular vesicles that have this red containing gene, this red expressing gene. And so now this mouse tumor is able to express red. So our next step is, can we use extracellular vesicles to deliver the MDM2 SSO? So again, we use the lentivirus expressing our SSO sequences, and we loaded these into the SSOs into the extracellular vesicles using this lentivirus technique, and we delivered it to tumor cells in vitro. And what you can see is that we see very nicely a nice induction again of MDM2-ALT1 after just 30 minutes of treatment in these tissue cultures. So our next step is to do this in tumors and to see how long this can last, how often it needs to be administered, and what is the efficiency and efficacy of this delivery treatment. But we're very excited about the possibilities of using these extracellular vesicles to deliver these SSOs to get induction of MDM2 alt splicing. So it looks like a very promising idea that indeed we are able to induce MDM2-ALT1 using these antisense oligos targeting just exon 11 here. So this SSO targeting exon 11 can cause skipping of all these preceding exons in the endogenous gene, not just in the mini gene, and reactivate P53 as I've shown, and activate P53 target genes. And hopefully we can use that to kill cancer cells either alone or in conjunction with drugs that I've shown here with these small soldiers that are helping the P53. So with that, I'd like to acknowledge the people in my lab that have done this work. Mostly Matthias Montes has been really integral and doing most of the work on MDM2. And Safia Khurshid has been the driver of the insulin receptor project. And this project was just published in MPJ Precision Cancer earlier this year, I think in January. And much of this work on MDM2 has been published previously with Aishwarya, and some exciting things with MDM2 are coming out later this year, we hope as well. And Akila, who's an MD PhD student in the lab, who is picking up splicing in Ewing sarcoma and going to start exploring different cancers. Hannah Ackerman, who is an undergraduate in the lab, who will be headed off to medical school next year. Claudia Kelly, who is a wonderful technician that was able to attract from California to come join the lab and become a splicing guru. and Anna Ivanovich, who is our administrative assistant, who has become a splicing guru and is no longer a layman, is becoming a scientist just by helping me read grants and submit my papers. So she's been wonderful as well. And of course, thanks to Aishwarya and all the other students in the lab that have come through, got their PhDs, and helped develop these projects along the way. Of course, I have to thank Ionis Pharmaceuticals for their help in developing the antisense oligos, as well as the many collaborators in my department and outside. And I would be much remiss if I didn't thank people who funded this work and made it all possible, from pediatric cancer foundations like Cancer Free Kids and Pediatric PCF, all the way up to the National Cancer Institute. We're very thankful for the support of the work. And I hope I left about 10 minutes for questions if we have any, and I'll just put the 2 genes up here that we talked about in case you have specific questions about these 2 genes. So thank you so much. I hope I kind of started talking fast at the last part because I realized I was getting close to time, and I have exactly 12 minutes left to answer questions. So I think that we hit the nail on the head there with timing. So thank you, and I'd be happy to answer questions.
Thanks, Dawn. That was really nice. And I do have loads of questions, but I feel like if I start asking questions, you're going to look at me and go, you should know that. But
of course not. I think there's never, I think you probably maybe make me think of things in new ways like you always have.
Indeed. I'm just going to take host privileges and ask you just one question.
Sure.
This is basically something that you didn't quite touch on the MDM2 part. So of course, what you didn't quite delve into is the whole paradox of MDM2 alt1, which was basically the whole premise of my PhD thesis. But one thing you did point out was that of the 3 SSOs that you wanted to use, you were looking at the ones that caused the complete switch to the alt1. I was wondering if, given these sort of paradoxical I'm fully cognizant of the danger of just slipping into my own PhD thesis right now, but we do know that cancer cells do express MDM2 alt1 as well. So if we try and switch into MDM2 alt1, what is the almost completely using the second 2 oligos? What do you think we might encounter the danger of actually exacerbating the cancer rather than helping it?
Well, I think that you have a good point, and I didn't get into the complexities of the MDM2 paradox, and that it can be both beneficial to have splicing in the case of damage and beneficial to have splicing in the case of cancer to activate p53. So I didn't get into that because of time, and I wanted to focus on the cancers that are overexpressing MDM2. But there are a number of cancers that overexpress MDM2 alt1, and those are cancer driving in the absence of p53. So we believe that in this particular cancer where MDM2 is overexpressed and p53 is not mutant, p53 mutant would be another story here. If p53 is wild type and can be reactivated, we think that it would be beneficial. And we believe that it won't be a double-edged sword because it won't be prolonged expression. We won't be turning this expression on permanently. And so normal cells are expressing very low levels of MDM2, so you're not in p53. So even if you aren't able to target well, you have a very low chance of this having global effects that are deleterious. And in the cancer cells, that expression of MDM2 is very high, and that you're hopefully targeting more with more higher expression of these SSOs. Activating of this p53 should cause cell death or cell arrest, or make them more sensitive to other cytotoxic agents that can't work in these cancer cells because MDM2 is overexpressed. So this would be analogous to Nutlin-3. So the drug that's used to reactivate p53 by getting MDM2 away from p53. And it would be more beneficial than Nutlin-3 because Nutlin-3 only acts on MDM2. So MDM4, a sister of MDM2, sister molecule, is still negatively working on p53 in that case. So Nutlin-3 can turn off MDM2, but it can turn off MDM4. But what I didn't mention in this talk is that our MDM2 alt1 turns off both MDM2 and MDM4, right? And so then you can really reactivate p53 in the absence of these negative regulators.
Indeed. It's lovely. Well, I think I should give way to other people's questions, indeed.
Sure.
Let's start off with Letitia. She wants to know how sure you are about the specificity of the SSOs, and can they be targeted to specifically to cancer cells and not to the other healthy cells?
So I think that's two very good points. How do we know that it's specific to our transcript of interest is the first question. And these are 21 nucleotides long, so they're not present everywhere. And we did a search, and they're not present in close to any other exons that are regulated. And we also looked so we did that bioinformatically, and that's all outlined in the insulin receptor paper as well as the MDN2 papers that we've done previously, how we were controlling for specificity. And in particular, if the insulin receptor paper, which I kind of glossed over in the talk, is we also included a non-regulatable expression of the insulin receptor gene that cannot be changed at the level of splicing. And we included that overexpression, then we didn't see the same change with ASO. So which says that the effect is basically due to the ASOs changing splicing, because when you overexpress insulin receptor that cannot be changed by splicing, you no longer see that effect. So that's one thing targeting as affecting to address the specificity to the transcript. Now it's addressing the specificity of what cells you hit, right? That's another area of concern. It can be problematic, except for that the cells are already expressing IRB, so it doesn't matter to them if you turn on more IRB. So that won't matter in the IRA example so much. There are some caveats to that. Some neurons express higher levels of A, which is confusing because they're not growing, so they must use A for something else. But luckily, the blood-brain barrier may work to our advantage in that effect, so that the oligos may not get to those cells. So that is something we need to consider for therapy. For the MDM2, I think we addressed that with Aishwarya's question. There's also a weird thing that we're seeing. We have no idea about mechanism, but these extracellular vesicles that we make from cells seem to home to cancer cells. We don't know why or how. This is not what you see when you make extracellular vesicles with nanoparticles. Those don't hone as well. But these made from cancer cells seem to hone to cancer cells, so that may be an advantage. We need to work out the mechanism of that. We have no idea how that's happening. It's sort of a black box right now. It's really fortunate if that is the case, but that might work to our advantage in the future. So good questions, thank you.
That also answers Hui Shan's question about how you direct the extracellular vesicles to specific cells. Right. So following up with that again, another question from Letitia. Oh, sorry. Another question from Hui Shan asking how expensive these treatments are, and is it cost efficient at all to make?
So the ASOs are really inexpensive to make. They can be. So I think that, but the delivery vehicles could be adding a price. I have kind of a vision of being able to take cells from the patient and use those cells to make, infect them with lentivirus, make this SSO from their own cells, and then inject their cells back into them, the extracellular vesicles back into those patients. So that's kind of a long-term idea of how this might work. And so if that's the case, it would be a little bit personalized medicine, and you could put those cells in the freezer, and if you needed to redose, you could pull those cells back out and get more extracellular vesicle and more ASOs, which would decrease the cost. I mean, as far as personalized, a relatively inexpensive method of personalized medicine, I suppose.
Well, that's very interesting.
That's obviously way in the future, and probably shouldn't even be talking about it yet, but I think could be a really cool idea.
But I agree. I think I think it might be best to try and characterize some of these vesicles anyway, to see whatever you're making from the cancer cells, what load are they bearing that's right, directing them quite specifically. Because that would be a quick question.
you're going to carry a lot of other things too, not just the SSO. So you have to be careful about that as well.
Yeah, exactly. So Letitia wants to follow up her questions by asking, how long do these SSOs stay in tumor cells?
So that's a good question. So the SSOs we get from Ionis that we can load in the nanoparticles, they're very stable. They're modified, they're stable. But the siRNAs we make from lentivirus are less stable because they are subject to degradation like all RNAs in the cell, right? They're not protected by these modifications. So that's another thing we have to think about carefully is we get a nice response in 30 minutes, but how long is that response going to be able to be sustained because of the degradation of these siRNAs, and how often do we have to redose? Or are there things we can do to stabilize those RNAs in cells? So that's a very good point.
Right, the next question is from Joshua, who wants to know, can some bacteria and viruses turn off and on some genes that are involved with tumor development?
Yes, they can. I'm not sure what the question is exactly. Can you say it again?
No, he just wants to know if there are some, if bacteria and viruses can turn on and off some genes that are involved with tumor development.
So I am not very familiar about bacteria, but I do know that yes, in the case of viruses, they do hijack. We're hijacking them for our purposes for this particular extracellular vesicle therapy, but they also, when they infect us, they hijack our genomic material and they hijack our cellular machinery to make their proteins, and they absolutely can turn on oncogenes and turn off tumor suppressor genes as well. And some of their genes cause cancer too. So some of the virus genes.
Antonia wants to know, could ASOS be used instead of the RNAi tech?
So I think that that's a really good option because they're modified and stable, right? But then it becomes a delivery issue. So we really need to balance the stability of RNA and being able to specifically target the cells we want to target. And that's really the walk we're trying to make right now, is to walk that balance between stability and delivery. And we really want both. But the two systems we have now, you either have stability or you have delivery, right? So we have to figure that out. So there's clearly lots of work to be done.
Thanks, Dawn. Are there any other questions from the audience? Yes, here's one. Hafsa wants to know, have you tested on people yet?
We're a long way from that. We haven't even tested these oligos in mice yet. So we are working on that, and we hope to get there at some point. We are working with a surgeon at OSU that happens to work on an adult cancer called dedifferentiated liposarcomas, which is a very small subset of sarcomas in adult patients that miraculously almost 100% of those cancers have highly expressed MDM2 and wild-type p53. So and they're very large tumors, so we can get a lot of samples from these patients. So we're hoping that we can really use that model to test this therapy. And we're working with Rafe Pollock at OSU CCC for that. So that's another exciting application of this event that we found in pediatric cancers that might actually be applicable to adult cancers as well.
Thanks, Dawn. That's amazing. It's actually really nice to see the way the project has come along. I mean, it's come so far. I'm really happy to see that, and very excited to see how this goes forth.
Thank you.
Yeah, thank you very much indeed. Hopefully we'll be able to invite you again for a follow-up talk, and that time you would have tested on people. So.
You might want to wait a while for that. We're working on it. Thank you.
Anyway, thank you very much. That's all the time we have for today. Do follow our website for more Live from Lucy events and other Lucy Cavendish College based events. Thank you again for attending today's talk, and thank you, Dawn, for joining us.
Thank you so much. I enjoyed being here. Thanks.
Thank you, and have a lovely evening, everyone.
And thank you all for the great questions.